Okay, so it's a pleasure to have Ariel from Plantfish talking to us today. Before we start the talk, I would like to mention that usually we meet in the TMC webinar once a month in the last week of the month. But because this week, next week, we have an event in Tel Aviv, it's called New Tech, and that's a 3D printing day. So most of us, or some of us, will be there. We moved the monthly seminar for this week. And as I said a minute ago, we have Ariel Skelani from the company Plantfish, and he's going to talk about today about 3D printing in biofishes, I guess. I don't know. Probably you're going to tell me.
Actually, I'm gonna talk a little bit about like what my PhD was, which was bioprinting of vascular tissues, and then I'll give some snippets about our company.
Okay, great. So the podium is yours.
Thank you so much. Okay, can you see my screen?
Yes, we can, but not in full screen mode. Now we do.
Awesome. So I'm really happy to be presenting you this mixture of my PhD studies and my current work. First of all, I did my PhD at the Technion at the Levenberg Lab, and right now I'm working as a CTO at the Plantfish company, and you'll hear about it a little bit more in a little bit. We'll start. I want first to tell you a little disclaimer. This is going to include some biology, but it's built in a way that you will able to like follow it as much as I can explain it well. So bear with me. I promise you at least fabulous confocal images. So tissue engineering is a science that aims to repair tissues in humans. The process starts from taking cells from a patient. I think, how can I do the little laser? It's Control L. Yeah. So you get the cells from the patient. These cells are cultivated in 2D until you get enough cells to create a tissue. Then you combine it with hydrogels and 3D porous scaffolds to give like the natural environment with the hydrogel and the mechanical support with the scaffold. And then you see them together, you let them mature until they express the features that you expect to see from the target tissue. And then once you have a mature tissue, you reimplant it into the patient. That's the basics. Oops, doesn't let me. Okay, doesn't matter. So tissue vascularization is critical. Like most tissues in the body have networks that give the cells their nutrients and the vital gases that they need in order to live, function, and proliferate. The transport of these gases and nutrients is limited by the diffusion limit to 200 microns. So whenever you have a vessel, all of the cells that are within these 200 microns can live. Those who are far away won't, because they lack the nutrients. That's why vascular networks are so important in tissues. When we look at the organization of the vascular networks in the body, we have a tree-like shape, which starts from the trunk, which are like the arteries. It ranges from microns to centimeters, and we go from the large arteries, then pass to the arterioles, and then we veins and arteries meet in the capillary areas, which are the tiniest of them all, and that's where the tissues receive the nutrients from the blood. The motivation for this project was to fill a missing link that it was in the bibliography, that we couldn't find anyone doing it. So it was very important for us, and you'll understand it right now, which is there are large scaffolds that are used like big vessels in the body, which are implantable, perfusable, and they have clinical use, but they don't have any kind of hierarchy and limited geometries. On the other hand, we have like the microscale hierarchical vasculatures, which go from big to little, like from small to a little smaller, which are perfusable and great vascular hierarchy, but they're not implantable. They only have limited geometries, and they're mainly a research tool. And also you have the microvessels in gels and scaffolds, which are like vessels are really tiny. They're implantable, they have great mechanical support and geometry versatility, but they're not perfusable nor contain a vascular hierarchy. So the main objectives of my PhD were to fill this gap from the tiny to the larger. And the idea was to create functional mimicking vascular bells and mesoscale vessels in the size of the millimeters using 3D printing technologies. Also, the next step was to connect between these vascular bells and the large vessel using the correct factors and cells that will allow that. And then taking all of this construct and implant it into a living animal to prove the feasibility of this technique. As many of you might know, 3D printing principles start from a real or an imaginary object that you 3D design and you create it into a specialized computer software. You slice it into thin slices that will represent a single layer of the object, and then you create this layer by layer deposition until you mimic the object that you design. But what about 3D printing? 3D printing has the same principles as any kind of printing, plastic printing, metal printing, but the difference is that you aim to create in vitro tissues with living organisms and biomaterials as building blocks. This means using hydrogels, biocompatible polymers, cells. And you can see here a little example where you can print a plastic that will give the structure. It will work as a scaffold and the hydrogel between this plastic scaffold. Mainly, this is not like a filament deposition, but it's based on cartridge and air pressure. And you extrude the material through a nozzle while it moves. And once you deposit the bioink is the term that we use to talk about like biological inks, you need to find a way to cross-link it in order for it to fixate and not to just go and turn into a liquid again, because the material in the cartridge is liquid, but it should hold its structure when it's deposited. So the advantages of extrusion-based 3D bioprinting is that you can use several cell types and materials at the same time. You can create complex structures which can be used to design patient-specific parts, and it might be automated and scalable process with reproducible results. The research approach was to create this hierarchical vascular construct that connects microvascular networks and mesoscale engineered vessels using 3D printing technologies. First of all, I'm going to give a little bit about like what does it mean to create a micro vessel network and how to behave in the micro level. So mainly when we create vascular networks, what we do is taking endothelial cells and support cells and mix them into a cell-friendly environment. As I mentioned before, you got the plastic scaffold. And when I say plastic, I mean like polylactic acid, which is a compatible plastic that can be reabsorbed by the body. And you also add an hydrogel that mimics the cell environment in the body. Oh, that's right there written. Thank you, Ariel. And then once you see them together into these scaffolds, they can they create these vascular networks on their own, because those are the things that they know how to do. That's why once you put them together, they'll start forming the vascular networks in which the endothelial cells are in charge of giving the tubular structure that we all familiar with. while the support cells give mechanical support and network stabilization, and also release angiogenic factors that will give the cues to the endothelial cells to form these vascular networks, and also give some cues for cell movement or migration, which is very important in the body. You can see here some confocal images of the tubular structure, which is formed by the endothelial cells in red. And on the right side, you see how the support cells surround and give mechanical support to the vessel that was formed with endothelial cells. Our first motivation was to understand the decisions made by new sprouting vessels and how they migrate in a 3D environment. This was a collaboration with Professor Georg Lahan from Michigan University, and we wanted to create a 3D system for clear observation of microvascular networks and that we can assess in real time. And that's very interesting. Assessing these movements in real time, it's very interesting and gives you a lot of information. And studying the endothelial and support cells behavior during the sprouting formation. And sprouting means when you have a vessel and how they start building another sprout that goes in a different direction, bifurcating from the original vessel. I hope this is clear. The way that we sought to check this was by taking different scaffolds with different geometries to understand if the environment gives some cues or have some effect on the vascular sprouting decision-making process. For this, we created a novel stepwise seeding technique in which we coated a scaffold with a material that allows cells to attach. Then we only seeded the endothelial cells and let them grow until they fill the whole scaffold. Once we had the scaffold full of our endothelial cells, we seeded the support cells. Both of them are critical for the vascular network process formation process, and always keep it in mind. You cannot have the vascular formation like this and stable vascular networks without both cell types. And once we see the support cells, we let them create vascular networks and we check how they behaved inside the pores of the scaffold or like the spaces in between. The cells were seeded in fibrin suspension, which is a bio ink or a hydrogel that gives them the capability of moving in 3D in all directions, actually. If we compare the stepwise seeding, we can see on the top row that once you have the endothelial cells, ECs, and then you add the support cells, you get very clear vascular networks which follow. You can feel how they follow the geometry of the pore. While when you do the simultaneous seeding, it's balagan. It's all everything is everywhere, and you cannot really have some conclusions of the sprouting decision process. When we take images of the confocal, using a confocal microscope of the vascular networks, we can see that they are actually hollow. That means that they're actually making vessels and not just like clusters of endothelial cells. We took these three shapes and we compared some critical parameters for endothelial cells and vascular formation. I'm not gonna go into details, but what is important is that they remain very similar in these characteristics, which are total vessel length and total vessel area, which means they don't have a significantly different behavior in regards of length and area. But when you look at the images, you see something curious. There's something interesting about how they distribute according to the pore shape. So we developed a MATLAB code in order to analyze these directions of the sprouts within the pores, and we got really interesting results, which saw that the circles have kind of an homogeneous distribution, while the hexagons and square shapes gave specific peaks of directions. And if we look at the shape of these pores, we can see this. The connection for the circular is like all over the place, And it makes sense because it doesn't really have specific characteristics in the shape. It's fully symmetric. While when we see the directions that connect the center of a side to an opposing corner, both from the hexagon and the square, they really match with the peaks that we got in the analysis. And this is one of the most beautiful confocal time lapses that I made, and I'm really proud of it. That's why I'm saying it. We can see the vessels sprouting of the endothelial cells. All of the colors are different, are all endothelial cells, but just expressing different fluorescent molecules, okay? So the red, green, and blue are all endothelial cells in real time, moving and migrating to the sides or to the corners. We did manual tracking of these sprouts for all of the shapes, and we understood that they have a high tendency to depart from the side center of the scaffold towards the corner of the scaffold. And that's a very curious thing to see because no one showed ever like this, at least not that I found. And you can see also that the circle presents like a kind of all over the place migration. So we thought, what is affecting this? What is it? What's going on here? Like, why do they go to the corners? What is so special about corners? And then we decided to stain for the smooth muscle actin support cells. Smooth muscle actin support cells means that they have a higher level of maturity as a cell that can give support to the vessels. So we stained for it and we checked the localization of this wall that you can see. It's supposed to be full of support cells. Even you can see the nuclei in the center of the pores, but they're not expressing alpha smooth muscle actin, which means they're not mature enough to provide the support that they need, and also not to increase the secretion of the angiogenic factors. And it's lovely because it matches the higher concentration on the corners and the lower concentrations on the side and the side center of the wall. This is a whole, like the whole contour, and the peaks represent the corners. And this middle distance between the peaks represents the center sides, the side centers. So we also noticed that the support cells exert compacting forces extremely. Like, it's hard to assess here because it's microscale, but this is a time lapse of the first 3 hours, I think. And you can see the beads that we injected into the gel, how they compact and go towards the center. On the right side, you can see the gel displacement vector field that represents the beads. And in conclusion for this first section is like this novel CD protocol allows for creating organized vascular networks and clear real-time imaging of the vascular phenomenon. Also, alpha SMA support cells and even distribution is a key factor behind sprouting decision. And also, alpha SMA support cells show a high preference for concave geometries, which is really interesting. And the support cells exert compacting forces in the gel. And the scaffold provided mechanical support during gel compaction, meaning that the gel is not completely destroyed, thanks to the scaffold that holds the whole tissue. Now to the thing that I think it's going to be more interesting to you all, is engineering a hierarchically vascularized tissue using 3D printing. The experimental workflow in this work started from creating a 3D printable mold, a 3D printed mold using a material that is water soluble, and we filled it with a PLLA PLJ solution, which is polylactic acid and polylactic glycolic acid, which is an absorbable plastic. That means that the body knows how to convert it into living tissue, and it doesn't generate rejection of these materials. It's very important that, because otherwise, if you will put a material that is not compatible, you will get a rejection and the tissue would not function as expected. Once we fill this PLLA, PLJ solution into the mold, we washed off the mold, so we received this tiny, nice vessel scaffold. Separately, once we got the scaffold, actually not separately, we took a dual head printing system and we separated it into two cartridges. We took two materials for printing. One contained the support cells and endothelial cells in a recombinant human collagen matrix. Recombinant means that it's not made in humans. I'll tell you a little bit more about this in a tiny sec. And in the other layer, you can see the colors match. In the other layer, we have the tissue-specific cells, because only creating vascular networks is it's good, it's fantastic as a investigating tool, research tool, but it has to have a purpose, and this purpose is to nurture a tissue-specific cell. And the material was cross-linked using a 405 nanometers light. Once we got the whole bulk printed, we took the scaffold, okay, and we placed it inside the main hole that had the vascularized tissue. And as we mentioned before, we saw that the supporting cells exert strong forces that compact the gel. So we took advantage of this phenomenon and let these forces to take our bulk and really tighten around our scaffold, which makes the construct a single unit and something that cannot be displaced. For example, it cannot slip out the scaffold. Once we had the compacted tissue, we coated the inner part of the polymeric scaffold in the shape of the vessel and created what is called an endothelium, which is exactly what happens in the body. In the body, we have our vessels are made of cells, and in the inner part where the blood is in contact, we have the endothelial cells that create this barrier that lets the blood flow without issues like coagulation or leakage. We then culture this for another week after we created an endothelium, as you can see in the little thing, in this little square. Sorry, can you see my cursor? Yes, okay, awesome. So we got the endothelium formation. This is the endothelium. It's a single layer on the inside of the vessel, and it should connect to the vascular network that forms spontaneously on the gel bulk. Once we got it mature and we verified that it actually flows from the center outwards, we implant this in an animal model, specifically in the rat femoral bundle, into the femoral artery, and we close it and we assess the functionality of that tissue. Like every other 3D printing technique, the material is critical. You cannot work without knowing the parameters of your material. So collagen, for example, is an excellent hydrogel for tissue engineering because it's the main structural protein in mammals. It contains cells attachment domains that let the cells attach and reproduce and function. The cells can also remodel the material, which is super important. It's a process that happens also when you have a wound. The wound is also like a tissue remodeling in order to close the gap of the wound. So it's critical. But it has a thermal cross-linking, which is very hard to control, and soft cross-linking material. It's the result. For you that you don't know, cross-linking means when you turn it from a liquid material that you can inject so into something that is stiffer and can hold its shape, okay? That's a very important aspect of bioprinting. These two last characteristics are actually not good. So we sought to collaborate with Coldplant, which they developed a collagen metacrylate that has improved characteristics for 3D bioprinting. First of all, allows for photo cross-linking, which means when you illuminate your material with a specific wavelength, the gel will almost immediately cross-link and create that stiff structure, which is fantastic for tissue engineering. And also it has tunable physical and mechanical properties, which is also excellent when you're trying to mimic a specific tissue. By study, we then perform various experiments in order to understand which is the best conditions for hydrogel or bioink, and we chose like the cell concentration, the photoinitiator that allows for the photo linking, and we decided to add a porogen, which means that creates pores inside the gel bulk. Hope that everyone is following so far. I hope I'm not going too fast, but if I am, I'm sorry. I'm doing my best. So testing the bioink means checking its mechanical properties. We need to assess that we have a viscoelastic behavior after photo cross-linking. And we can see on this graph of the modulus versus the time that the moment that we turn on the light, the photo cross-linking happens, and the storage module is way higher than the loss modulus, meaning that it's a viscoelastic material. Also, the elastic modulus of this material is around the 30 kilopascals, which is relevant value for engineering human tissues. For example, muscle tissue has approximately 30 kilopascal elastic modulus. We also wanted to see, okay, the material is fantastic. Can I print it? The answer was yes, I can. You can see it on the left, the design. That's the Levenberg Lab logo, which it's where I did my PhD, if I didn't mention it yet. And on the right side, I took 2 heads with different beads with this collagen, but instead of having cells, I just put fluorescent beads, and I printed the L in red, the other L in blue, and you can see that it's a good approximation of the original design. It's important to remember that the position of bioinks is not as good, not as perfect as the position of plastics. It's very important, and if you have questions about it, you can ask me at the end. Well, now that I know that how to make vascular networks, and now that I know that I can print the materials, we need to check both of them together, right? So what I did was taking the shape that you can see on the right side. That's the actual print. You can see a scale bar here, it's a 1 millimeter, and that's the wave pattern inside. And in green, in this little thing, you can see the endothelial cells. But for day 0, looks like little circles because the cells are still overcoming the trauma of being printed. But by day 3, they already start showing signs of creating vascular networks. And by day 7, we get a fantastic vascular network all over the place. And what I mean all over the place, not only in the XY, but also in the Z axis, because you can see here that the image is taken from a confocal microscope, and the top cells appear in blue and the bottom cells appear in red. That means that at least we have a connection of these vascular networks through 500 micrometers, which is twice the diffusion limit, which is over twice actually, which is very nice. But as we talked before, 2 things that we talked before. First of all, people are trying to create perfusable vascular networks using only gel bulks that contain a hole and let them form vascular networks and wish for the best. But we also understood that the support cells that help create vascular networks exert really strong forces of within the gels that have a compaction that might ruin the original geometry, right? That's what we are seeing here. like for example, I'm looking at the perfusible area of the bulk, and by day 1, it's already losing most of its surface. By day 7, you tell me where the hole is because I cannot see it. And this is, like I said before, a reminder: support cells exert high forces. And the conclusion that we jump to is this would be impossible to perfuse. If we want to implant it, this is gonna be impossible. I'm not being extreme. It's gonna be almost. I'm gonna give the doubt, the right of the doubt to this one, but it will be at least almost impossible to perfuse and implant it into an actual vessel. So we thought, okay, let's create a scaffold that will provide the support, the mechanical support to the vessel, to the bulk material, and also will allow us to connect it into a real vessel and perfuse the material. So first of all, we took, as I said at the beginning, we took this material, which is called BBOH. It's kind of behaves similar to PVA. You can print it like PLA, but you can wash it off after it's done. So we have this structure that has an external structure as a vessel, but inside you have a little rod and tinier rods that will create fenestrations on the wall of the final scaffold. You'll see it in a bit. Okay, it's not working. Why is it? Okay, yes. So I filled these these molds with PLLA, PLGA in a dioxane, which is an organic solvent. That means the BBOH, the water-soluble material, won't react to it, like in compared to PLA that can be dissolved by dioxane. And using centrifuge, we took the material inside, we froze it, we did the lyophilization to evaporate only the solvent while keeping the shape of the polymer, and then we wash out the mold. Once we wash out the mold, we got this cute little scaffold with a main channel and fenestrations on the side. You can see the mold on the right side, the scaffold on the middle, and a 1 shekel coin as a reference. When we look at the scaffold using an electronic microscopy technique, which is, don't remember, SEM, not TEM, I don't remember, scanning, okay, cool. We can see on the side view, the fenestrations are there. They have a very approximate size to the little rods that we printed on the side. And we also have this nice main channel that in which we'll create the lumen coating with endothelial cells. Lumen means, for those that you don't know, lumen means the hollow space within a vessel. Then we see that, like I said before, I wanted to create this endothelium to actually mimic and connect to the microvascular networks. So we see the endothelial cells on the inside. You can see I did a negative pressure in order not to have leaks on the external wall. And we have the inner part of the scaffold coated with endothelial cells that you can see they have this structure. Each of these green lines is the contour of one endothelial cell. These recreate this organization that is actually the one that you can see in an actual vessel from an explanted tissue. The coloring is what lets an endothelial cells attach one to another. And also, we were very happy to see that the endothelial cells weren't just there, but also they gained the actual functionality that we expect to see in the body, which means working as a barrier that doesn't allow materials to randomly diffuse, but it contains them inside the vessel. So if we look here, we took fluorescent albumin. Albumin is a protein that it's in the blood stream. And you can see that when we don't have the endothelium, there's a huge diffusion outwards from the center of the vessel, while the endothelium one almost has no leakage outside. You can see this in this graph the differences between the diffusion networks from the two cases. The full construct assembly, as I mentioned before, we put the scaffold inside the 3D bioprinted vascularized tissue. And on the right side, you can see the CAD design, which is a computer-assisted design. So the design is redundant, sorry for that. And this is how it looks right before we let it culture, which means just out of the printer and out of the assembly line, let's call it that. And as we mentioned before, forces exerted by these lovely support cells, they really compact the material. You can see it in a 16-hour time lapse. If we didn't have the scaffold, we would have no lumen of the bulk gel whatsoever. In green, you can see endothelial cells. We did, again, the back, once we assemble it like this, that we have the scaffold and the bulk material on the outside, and we let it compact, we see the endothelial cells with a different color. You can see down the legend, we have the gel microvasculator endothelial cells in green on the outside, and the scaffold endothelium cells, which are supposed to create the endothelium in the inner part of the scaffold. And then this is a side view of the whole thing, but when we cut it in the middle, we can see how lovely and they're distributed all over the thickness of the scaffold, the gel, and also in these little arrows mark the endothelial cells from the inner part of the scaffold migrating through the fenestrations that we purposely create. For this reason, to let them communicate in a direct way. If we take images from the immediate part outside the scaffold, we can see that in this confocal image, that endothelial cells from the endothelium, which was inside, they really connected and formed, they collaborated to create one connected vascular network going from the big scaffold with the red cells to the small vascular networks with the green cells that were inside the printed gel. This is really nice to see, in my opinion, always my opinion. But are they really connected, or was just like a geometrical thing that we only can see through the images? The answer is they're really connected. How we checked this was by perfusing the large scaffold. We had a full construct. The large scaffold, we connected a pipette and we perfuse inside a solution containing micro beads with a fluorescent color. And when we see where the fluorescent beads are located, we can see they have a almost identical distribution with the vascular networks. You can see there the overlap of the two colors. Also, we check for the maturation of these vascular networks, which is important to assess the functionality of the vascular networks, because having vessels is not enough. They also have to be mature. And when we look at the gel microvasculature and the scaffold endothelium, we can see the presence of what we mentioned before, the alpha SMA support cells, that are the support cells that give mechanical support and release angiogenic factors, and the presence of this marker inside the gel speaks about a mature vascular network. The next step was, okay, with this everything in the lab, a Petri dish don't really need vascular networks. What will happen if we take it into an animal model? Okay, so the animal model, because of the size we were working, the ideal one was taking a femoral rat bundle, resizing that femoral artery, and implanting end-to-end anastomosis from our construct to the artery. And this was done by two of the most skilled people and nicest people to work with, much that works still. He's doing his PhD at Shulamit's lab, and Dr. Aidan Bredensky, who also finished his PhD at Shulamit's lab, and they perform all of the surgeries. I have nothing to do with this, okay? It's not me, it's them. I only did the in vitro part. So they did the surgeries, which are actually very complex, and wait, why do you do this to me? Okay, that's the shape, the gross shape of the construct. And once we have clamps here that don't let the blood flow, the second that you remove those clamps, you can see the beautiful flow of blood through the main vessel, which is very encouraging. Also, we did Doppler in order to assess the actual flow of the blood flow downstream or scaffold to see if it created problems like coagulation or some kind of blood detour that doesn't let the blood reach the feet. And when we implant or construct, we can see the blood flow in the feet, in the treated leg. It's really close to the healthy leg, which is the opposite side. While when, as a negative control, we just made a little ligature in the artery, avoiding all of the blood flow through it. And we can see that the perfusion is close to none. Then we wanted to understand what happened after implantation. So we let these tissues mature inside the rats for 14 days. And we took the tissues, we explanted them in order to check them out and see how they develop inside. And we can see these are explanted tissues. This line represents where the scaffold is supposed to be. And this part represents the printed gel, okay? That's important. That's a crucial point. Because, come on. Yes, we can see something very nice, how the host vasculature comes out from the inner part of the scaffold, which is the lumen where the endothelium is, and communicates with the external vascular networks. In red, we see the laminin, but laminin represents the vasculature of the host, that you can see here. And they have varying sizes of vascular networks, meaning that they do create a hierarchical structure, because we started from the millimeters and we are already gone into the micrometer range. If we look at what kind of vascular networks are formed, we can see that the tissues there have actually real life structures that are arterioles and venules, and you can see them on this right side, like when you have this endothelial, this vessel formed with endothelial cells coated with support cells expressing smooth muscle actin, it means it's a kind of artery, but smaller, so we call it an arteriole. One of the questions that pop up a lot in our head, in our discussions was, are the fenestrations really necessary? So, because most of the state-of-the-art implantable large vessels do not contain these side fenestrations. And this is one of the novel parts that we add to the process. So we wanted to test the effect of the fenestrations in the actual tissue formation. Once we implanted these same constructs with and without fenestrations, we could see that the gel vessel coverage was way higher than the one that contained the tubes with no fenestrations. And also interestingly, we can see that the size distribution of these vessels for the non-fenestrated, because they can't do through the porous wall of the artificial vessel, the scaffold, they do cover the gel, but they only can create tiny vessels, while the fenestrated scaffolds allow for larger vessels in the implanted gel. This is in the arterial ranges, and this is in the capillary ranges. Capillary are the tiny ones, arterial are the bigger ones. We needed to test it once again using perfusion. We took a contrast material that can be imaged using micro-CT, computer tomography. We injected this through the rat's tail, not even in the actual area, but through a rat's tail. That means that it flowed, I guess, through the whole animal system. And once it reached our implant, we can see that it was also in the center of the scaffold lumen and also in the vessels that are located in the outer part. The scaffold is this area that has almost no vessels. And on the outside is a printed gel. If we can see here, this, my friends, is the fenestrations. That's why they are so important. And for last but not least, we also proved lectins that they have a high affinity for endothelial cells. Again, we injected this from the tail of the rat, and we can also see all kinds of vascular networks inside the printed gel. But like I said before, endothelial cells, vascular networks are not enough. You need to prove it with a different tissue type. And in our case, we chose to create cardiac muscle tissues, which are derived from pluripotent stem cells, okay, induced pluripotent stem cells. It's good to know that the cardiomyocytes do not proliferate in vitro, so you cannot take a sample from a patient and reproduce it on a plate. That's why you can take induced pluripotent stem cells that they have the capability to differentiate into any cell type that you can think of and make them turn into cardiomyocytes like we did. And then we printed them next to a vascular networks, and we checked its functionality of the cardiac tissue. Like you can see, like the drawings before, you can see one layer is endothelial cells with support cells, and the other one is the cardiomyocytes that were printed in collagen. And we can see this beautiful migration from the layers of the vascular networks parts towards the cardiomyocyte parts. Cardiomyocytes here are in red, you can read it, but I have to say it. And you can see the beating of the tissue. You see these green waves are the synchronous beating of the cardiomyocytes after printing, which in my opinion is just beautiful. You check it by seeing the calcium flux within the cells. And we also wanted to see, yeah, they beat, but do they actually behave like the real thing? And the answer is yes. How did we check this? We check this by taking a common known drug that increases the beating rate and measure the beating rate of the engineered tissues. So you can see this is the best baseline when no drug is added. And once we add the isoproterenol, we have an increased beating rate. But again, does this end in a tubular shape that only will be very specific for certain areas of a wound? I wasn't satisfied with this answer, so we sought to expand this concept into patient-specific vascular constructs. What this means is taking a CT image of a human coronary artery, creating with the same materials we talked before, the one that's it's water soluble, creating the patient-specific vessel scaffold, and then hydrogel 3D printing the cells around our nice little scaffold that mimics the one from an actual human patient. You can see in this video how the construct was created. The red is the hydrogel. that is creating layer by layer the full bulk of the material. And you'll see that it will start creating areas in which doesn't print, and those are the hollow spaces in order to put our special form scaffold. You'll see in a sec. Now we put the scaffold and continue the printing process on top of the scaffold, which means that we can achieve a fully surrounded vessel with biological materials. We check it from the bifurcation outlets. We can see that it's connected in between with the gels. The front view is also everything is coated. And for the conclusions about this work is that the recombinant human collagen metacrylated bioinks can be tuned to support tissue formation, and also that the polymeric vessel scaffolds provide mechanical support for bioprinted tissues. We also use a novel approach that we use to achieve hierarchical vascular constructs that communicate mesoscale vessels to microvascular networks. They can be implanted directly into the host vascular tree and promote construct host tissue integration that we saw when the host tissue, the vessels went into the 3D printed gel. The host vasculature recapitulates the vessel hierarchy within the implanted hydrogel. This means that we actually saw arterioles within our implanted gel. Also, the engineered vascular tree can nourish IPAs like induced pluripotent stem cells derived cardiomyocytes, maintaining their functionality in vitro. And too long didn't read, using the 3D bioprinting, we develop a unique methodology with the potential to create personalized hierarchical vascular tissues for implantations. All of this has been published in a paper in the journal Advanced Materials. It's open source, so you can check it. You can always ask me questions about it. And now I'm gonna move forward to section 3, which will be a little snippet about what I'm doing right now, which is creating plant-based salmon fillets using additive manufacturing technology. So a couple of words regarding 3D printing of food. 3D bioprinting and food printing are super similar. So similar that people are even doing culture meat 3D printing, which means doing the same that I did for my PhD, but instead of having the goal of creating tissues for implant, you create tissues to have a very nice steak. So the medium to low viscosity of the materials is very similar between the two approaches. Both of them need to be fixated, the materials after deposition. Both techniques, because it's based on 3D bioprinting, so both of them can create complex structures that mimic real-life counterparts like vessel networks or a steak with fat. And it allows multi-material approaches, increasing the technique applicability and flexibility. Again, in my project was vascular networks and cardiomyocytes. In a steak can be fat tissue and a muscle tissue to mimic a steak. But it has its benefits. You can create personalized meals. You can create unconventional, you can make people eat unconventional food. For example, plant-based steaks. For people that don't want to eat plants, but they do like steaks, you make them in the shape. Don't forget that food always comes fresh from the eyes and the nose and the sensory system. So you have to convince them using something that it will remind them of the food that they like, and also it's repeatable and reproducible. However, it has drawbacks, which are that the print takes too long per product. 6 layers can take, a full thing can take up 2 hours. It's very hard to scale up 3D printing as we know it, and the consumables and trainings are very expensive. So we need to develop a novel additive manufacturing technologies to overcome these drawbacks. And that's when I want you to meet Plantish. Plantish is a company that aims to create full salmon fillets with plant-based materials. I'm not going to be all preachy right now about veganism. That's not the point of this. I just want to show you my motivation, or motivation, sorry. The motivation is that a lot of people on the world rely on fish as a source of protein, the main source of protein. And 90% of the world marine fish stocks are overfished, leading to insufficient supply to meet the growing demand, because people are reproducing while fish are getting unreproduced. Also, in today's fishes, you can find toxic mercury and microplastics and other chemicals that contaminate the ocean. Actually, a couple of months ago, they published a paper about first time of microplastics being found in a person's bloodstream. Okay, that's scary. And also, fishing gear alone accounts for 2 trillion pounds of pollution annually. Like all of the nets that you use to gather these fishes, yeah, those end up in the bottom of the ocean, which is kind of ironic. So the global seafood market is one of $586 billion, but the consumption of this fish is 80% comes in whole cut presentation, like salmon fillet, and 20% is minced meat, like for example, tuna in a can. And currently, there is no fish whole cut alternative in the market. So creating whole cuts, it's rather harder than creating minced meat, because minced meat doesn't really have a clear structure. You have like a bulk of tasty something, but it doesn't represent like any kind of tissue. And for example, a fish has a complex 3D muscle, fat, and connective tissues arrangement. Also, the texture is derived from fibrous materials, right? And the plants are not fibers. So you need to mimic in some way these fibers that you wanna create in your product. And also the scalability of producing whole cut fish at scale is hindered by the desire to get realistic structures. So if we will take conventional 3D printing, it will take, as a guesstimation, maybe 10 hours printing. Between health changes, you take one material, then the other, it's gonna be long. And you are hungry right now, knowing 10 hours. So we are developing a breakthrough additive manufacturing technology to produce plant-based whole cut fillets at low cost and massive scale. Since we are still developing the technology and it's not yet patent protected, I won't be able to give you details, but stay tuned. In the future, I'll be happy to share with you our knowledge, because we also want other companies to find a way to make it faster, tastier, and easier. And now please say hello to the plant-based salmon. It's high in protein, it's rich in omega-3, has no mercury, no antibiotics, and no plastics, and is actually very tasty. What you're seeing in that image is our actual product. We were featured in many news, divulgation, and social networks, newspapers. This is Nas Daily. I think he has a lot of followers, so that's kind of cool. And we have really positive response from the people that try it so far. And why salmon though? Salmon is the most popular whole cut fish. The global market, salmon market, is estimated at 50 billion. And the salmon is a premium product when compared to other proteins and fish, okay? So kind of close to the finishing line. So we got the founding team, our lovely CEO, Ofek, who work as a CEO at Vegan Friendly. He's so charming that he'll convince you to invest in our company. We got Dr. Ron Sigsik, who's a veterinarian, has a PhD in veterinarian. He knows a lot about fish, and that's very important when you want to mimic some salmon. We got Dr. Ilah Helimelech, which is she's also a co-founder and the R&D director. She has a PhD in chemistry, and it's a former VP materials at Nano Dimensions. You got me, which I did my PhD at Shulamit's lab, and I am the CTO at the company. We got Mr. Eli Sinal, who is a food technologist that was the CTO at Nuva. And we got Eyal Briller as a founding board director, who worked as a former director of product at Impossible Foods. The new seafood competitions mean, when you look at it as a manufacturing innovation and the whole cuts, like how far we are leading in the area of manufacturing innovations and the whole cut structure in comparison to stuff that mimic minced food and only develop recipes but don't have an innovating mechanism to create the food. So our vision is to be the world's leading fish brand, alternative fish. And at some point, I wish fish at all. Thank you from the Plantish family. This is our current team, but everyone is super critical in the day-to-day basis. They all work so hard, and it's a lovely, I put family because it's such a pleasure working with these people. It feels that we are all oriented into creating this salmon to make it happen. So I'm really happy today. And also, as my last flex, this is the cover that we got from the paper in Advanced Materials. It's a cover that I designed, and that was my highest aspiration during my PhD, was getting a cover published, and I achieved that. And thank you all for listening.
Thank you, Ariel. We are a little bit late, but we have time for a few questions. Quick one, definitely, if you have one. Okay, maybe I will start then. Regarding the design of the arteries, again, many of us, including myself, are completely out of this area, but I'm kind of trying to understand the global picture. So you can 3D print the arteries, but you cannot really print all the capillary vessels in the process. So the vision is to do a two kind of stage of process. You're going to print the arteries and then let somehow the body or the host grow the rest of them. What's the vision?
The vision here is to actually create some kind of printable material that doesn't contain organic solvents and mimic this tube that we created using the mold, but to print it at the same time that you print the vascular networks on the outside. So it should be a one-step process. You print also the engulfing material on the outside and also the main vessel at the same time without losing
but
timing.
You cannot do it all the way to the capillary level, can you?
No, the position of cells is not that relevant in my opinion, because cells tend to migrate a lot. So even if you put it at this place, you can find the cell one day after 500 micrometers away from it, or 50, doesn't matter.
Okay, so again, I'm asking about the vision. The idea is to make the big vessels, 3D print them, and then let the body or the host grow the capillary vessels?
No. You also print on the outside, like I did here in the outer bulk. You also print the vessels that will create the vascular networks that will make the tissue nurturing much faster than if you don't add these vascular networks and let the vessels come only from the host, because that will be dependent on the velocity of the cell migration towards the gel, in comparison that if you already have these formed vascular networks from the Petri dish, and then once you implant it, you have immediate perfusion of the whole tissue.
You say immediate, I mean, is this taking an hour, a day, a month, a week?
Which part?
The process of building the entire network.
It takes 1 week.
About a week.
Yeah.
Okay.
1 week.
Any other question?
What is immediate is the perfusion of the blood within the construct, and that's critical.
Okay, any other question? Okay, if not, thank you very much, Ariel. Good luck.
My pleasure.
Let us know when the salmons are going to be in a supermarket near us so we can actually try them.
Stay tuned. I hope really soon.
We will. Thank you very much.
Thank you for
everybody,
inviting
and
me.
see you next month.
Bye-bye.
Bye-bye.